I am Bas Neggers, CEO of the spin-off company Brain Science Tools BV and guest lecturer at University Medical Center Utrecht. My lecture is about our concurrent TMS-fMRI approach of probing networks from the dorsolateral prefrontal cortex to the subgeneral cingulate and their role in depression. Most of the research that I'm showing you today has been published in 2018. The first author was Mr. Vink, also in the Symposium, and Mr. Petrov, myself, and others in human brain mapping. Uh, well, first I'd like to introduce the use of MRI for TMS in general. In the past, magnetic stimulation of the brain long ago and in its current form, initiated by Mr. Barker in 1985, was done largely blind by picking a location on the scalp rather than using knowledge about the interior of a patient's head. Today, many of you applying TMS know very well that acquiring an MRI image and determining or a functional MRI or even motor maps will allow you to more accurately target a region in the individual patient, thereby improving treatment efficacy. A little disclaimer here. So I am also, we as a company are also manufacturing and marketing the CE marked and FDA cleared Neural Navigator, a system for accurate MRI guided TMS. So I'm not completely neutral, of course. So that's the offline application, making an MRI, and then after that, using that MRI to place your coil. These days, however, we can also do online functional MRI and TMS at the same time. So applying TMS inside an MRI scanner with an MR compatible TMS coil. The promise here clearly is that in vivo insights in the networks engaged by TMS in health and disease might be able to allow us to individualize TMS treatment. So in major depression, RTMS treatment or repetitive TMS at larger frequencies is used to treat depression over the course of 6 weeks and almost daily treatment. It is possible to reduce the major depression symptoms by stimulating the dorsolateral prefrontal cortex. This is quite effective and now also reimbursed by most health insurers. But the conventional practice is still doing this blindly with the so-called 5 centimeter rule in front of the motor cortex, so no MRI used. Often you miss the target when you do this. That has been shown in quite a few studies as shown here. And Paul Fitzgerald showed in 2009 that when you do use MRI that you can increase the efficacy of your treatment by 30 to 50%. That's quite impressive. What about the clinical use of concurrent TMS MRI in depression treatment? So let us show you what we tried here with our novel setup. The nice graphics are courtesy of Jort Vink. He's really talented in making the nicest 3D renderings for presentations I've ever seen. So let's have a look at the brain in major depression disorder. Abnormalities in the major depression circuitry involve the dorsolateral prefrontal cortex, but also a region below the cingulate, the subgeneral cingulate. There you see a decrease of metabolites. It's also the site where deep brain stimulation is effective in markedly reducing depression symptoms. Of course, that's highly invasive and risky, but there seems to be two regions in the brain that are important in reducing depression symptoms. A famous study by Fox et al. Showed that the more effective sites from literature, they didn't actually treat in the study, are accompanied by a larger anti-correlation in resting state functional MRI between subgeneral cingulate and the dorsolateral prefrontal cortex on the surface that's reachable with TMS. So this led them to speculate that this might be it might be possible to select patients based on their intrinsic anti-correlation and by doing so improve treatment outcome. So this has been really a study motivating a lot of others, including myself, to do larger trials. Interestingly, the Fox group himself, Sadiki et al., could not show a relationship of anti-correlation with treatment outcome in a more recent study. However, they could show that when the spot on the cortex most strongly anti-correlated with the subgeneral seed region was more effective in reducing depression symptoms. So that was interesting. The negative correlation by itself did not predict treatment outcome. Something similar, Elaine Hopman and myself, and also Professor Yutse, who is present here today virtually with us in the symposium. We could not confirm that. We couldn't confirm either the original findings of Fox et al. Or their prediction that the anti-correlation between the subgeneral region and DLPFC predicts treatment outcome. We did find some more posterior regions who actually did show that property, so that's interesting. No one disagrees that the subgeneral is an important site in the reduction of depression symptoms, but it's not as easy that the anti-correlation predicts treatment outcome. The site, however, you could very well pick using anti-correlation mapping, so it's a bit more subtle or complicated than initially thought. Still very interesting. However, we at the moment do not know yet whether the stimulation of DLPFC even reaches the subgeneral. That's never shown. Is it a network that's actively connected? Is activity that you want to bring to the subgeneral region for major depression symptom reduction even connected? So our own DTI tracking failed to show a direct connection in humans even. So the picture might be even more murky. So what do we do to investigate whether stimulating DLPFC even is able to relay activation to the subgeneral cingulate, as is often assumed in attempts to individualize treatment using functional connectivity. So that's a premise that definitely needs to hold, right? So is it possible by stimulating DLPFC exactly MRI guided to activate DLPFC? So that's what the goal of our study was here. Right. So we did concurrent TMS-fMRI with a single pulse, during which we recorded functional MRI. If this works, it might even be possible to use such concurrent TMS-fMRI sessions to predict, to find the optimal site to activate the subgeneral cingulate. That would be really cool if that works. Fiddle around with the coil, move it, and stimulate until you find the spot that actually activates the subgeneral regions optimally. That's a cool promise of this in the future. So we used a setup that has been described before by De Weijer et al., where we used a MRI compatible TMS coil and a special setup. It's quite intricate, and electronics we devised ourselves to synchronize it all. Of course, you have to pay attention to Lorentz forces, so you don't want to have the coil too much away from the B0 field, because otherwise it could hit you like a propeller. And also you have to be careful to not stimulate during EPI readout. So we build in an additional break during which we either stimulate it sometimes or not, allowing us to capture the TMS-induced bolt responses of individual TMS pulses. We've published multiple studies about this technique. So we only did a small proof of concept study with 9 healthy individuals, volunteers. We interleaved 115% motor threshold with 60% motor threshold to the best of our abilities, cancel out sensations and sounds. MRI guided neuronavigation was used prior to putting someone in the scanner for placing the coil, but we did monitor coil position during stimulation. So this is how the setup looks. We use flex coils, allowing the subject to tilt the head so that the coil could stay perpendicular to the zero field. And with neuronavigation, we marked the spot directly overlying the LPFC, anatomical DLPFC of this person, on a cap. And we placed the coil there as good as we could before putting the subject in the scanner. So that's how it looked. Thanks again, Jocht, for the beautiful images. We identified DLPFC using a trick counting gyri. That's a well-known method to find the more anterior portion of DLPFC. Well, to the results, we could indeed demonstrate, and that was the cool part here, that not only we stimulate DLPFC or surrounding areas, but also for about half of the subjects, as you see here on the upper left, and also at the lower right. There we could clearly see evidence of activation in the subgenual cingulate during stimulation of the DLPFC. So this is the first evidence that indeed activating the subgenual cingulate is possible when you stimulate DLPFC. So but it's a really small group. So interestingly, that wasn't the case for everybody. We failed for a similarly large group of individuals. So that's interesting. This was not due to the exact stimulation point we ended up stimulating. So as we said, we used a method to anatomically determine DLPFC. However, of course, we didn't have a robot inside the MRI that could move the coil when the subject moved the bit, so it didn't always work out. So we thought, hey, maybe that predicts what we see here, but that was not the case. So we stimulated a relatively broad region in the frontal cortex, and then subgenual or not. So concurrent TMS-fMRI can be used to visualize network activity in response to TMS. TMS-induced activity can propagate to the subgenual cingulate. That's the good news. It was highly variable. Could that be due to functional conductivity? That's not clear. This might be useful for tailored TMS RTMS therapy. So if you can find the spot somewhere in the frontal cortex that most strongly activates the subgenual cingulate, this might be the best site for depression treatment in the subsequent weeks. So that is the promise here. How are we continuing? Well, luckily we got funding as a company actually, together with UMC Utrecht and 10 other companies and institutes worldwide, led by Philips Medical, to improve this technique. It's called SIGNET. We're very proud to be part of this recently started consortium. Here the first face-to-face meeting happened in Barcelona a month ago. The project is about image-guided neurological therapies in general. So it's not just TMS, but also MRI-guided robotic biopsy of the heart and other techniques. So it's really about the tech here. And our deliverable is a brand new concurrent TMS-fMRI setup. We'll be assisted by the experts in BEST at Philips Medical to build this, and some of our other business partners. We are going to build and are already building real-time guidance and tracking of the TMS coil inside the bore. So here we can actually track the coil inside the bore and correct the position if needed. So we have real-time access over exams. We can start, stop sequence, adjust angulation and so on, allowing us to minimize artifacts. We have real-time access to the data as it comes in for visualization of what we're doing. We are building a brand new MRI compatible TMS coil and controlling electronics. We will do visualization and validation in the scanner of TMS-induced currents for appropriate dosimetry, making the resting motor threshold finally obsolete. And we'll integrate that into our Neural Navigator product. So that's what we're attempting right now, and I'm really happy we found the generous funding from the EU to continue this endeavor. So we hope really to expand this proof of concept study to something that's clinically useful and also available with CE marking and FDA clearance. Thank you for your attention. I'm ready to take questions now. And my apologies for my voice. I'm currently recording this a month prior to OHBM while suffering finally from COVID. I'm recovering, but my voice is still a mess. My apologies for that.